All right, welcome everybody. Thank you so much for attending our Build a Cell seminar series. And thank you very much for Karen, who's going to be presenting today. Karen, I will let you introduce yourself and walk us through what you've been working on.
Great, thanks so much. And thanks for having me. So I'm in the Department of Chemical Engineering at Imperial College and also in the Center for Synthetic Biology. And really our interest in cell-free protein synthesis systems is in their ability to make proteins. So we're interested in these eukaryotic systems as a way of making proteins with post-translation modifications and things like that. But I've tried a little bit to try and map it a little bit for people in Build a Cell, and I'm also happy for people to ask me questions about things at the end. So for those of you who aren't as aware of cell-free protein synthesis, it's basically using the internal contents of the cell in the absence of the cell wall, just as the synthesis machinery. So it enables you to do things that are kind of midway between a biological and a chemical reaction. So proteins can be made, the ribosomes are there, amino acids can be charged onto tRNAs, you can have some basics of metabolism and things like that. But because the cell wall isn't there, you can put in things that you normally wouldn't be able to get across that barrier. You can top things up, and also energy isn't diverted as much towards the cellular processes, although some really interesting work has looked at the amount of ATP that's being used up, and it is going somewhere, often not into the protein synthesis. And compared to in vivo systems, they're a lot more well-defined and simple to use, particularly if you use things like the pure systems, which consist of purified proteins. And I know that's what Kate has used quite a lot in her work in the lab. So in the spirit of Build a Cell, what are the uses of this in the kinds of work that you might do? Well, essentially, it's providing you the machinery for expression of proteins in situ. So if you wanted to have sort of sensing and responding systems that you're setting up, where you've got like some kind of signal is being transmitted, something like that, you can have it be triggered by the kinds of things that would trigger it inside cell biology, or unnatural systems triggering it. You can also incorporate non-canonical amino acids or XNAs or things like that into it very easily because the cell wall isn't there. I mentioned that we're interested in it from a protein biomanufacturing standpoint, and for us, we really see it as a way of getting synthesis of proteins relatively quickly on demand. So imagine having an extract that you stock in the freezer along with your new DNA of interest. You can sort of mix those two, and in about 3 hours or so, you can have a bit of protein to sort of work with, play with, and see whether it's the protein that you'd like to invest your time in manufacturing. I've also mentioned that it's very controllable, so you can add things like chaperones pretty easily. You can add cofactors and other things that you would like to much more easily than you can do with cells, where you have to put it into the culture medium and then rely on transporters to get across the cell wall. For us working in the protein manufacturing space, it's also nice for producing those proteins that a cell really isn't very good at producing because it's cytotoxic. So think of things like endolysins or stuff like that, where the natural activity of the proteins is to kill the cell, often trying to produce those. In a cell-based system, the yields are limited for relatively obvious reasons. So in a cell-free protein synthesis environment, you don't have that worry. And difficult to fold or challenging proteins, again, a lot of times those will aggregate inside a living cell and cause, stress pathways to be activated and cell death and things like that. And you can avoid all of that using the cell-free system. And then some work that we've done looking at protein assembly, you can manipulate the reaction conditions to enable assembly. So you can change the redox conditions, you can add things like that as well. So there's a lot of really nice potential uses of these systems for manufacturing as well as for building a cell. There are still some challenges, quite a lot of them actually, and I would say the top three for me are that there are definitely lower yields in cell-based systems. So if you think about it as a manufacturing platform and you try to talk to, for example, biopharmaceutical manufacturers, the yields are a lot lower than they are with cell-based systems. And a lot of that is to do with dilution of critical components. So it's about tenfold or so more dilute than the inside of a cell, which is actually quite crowded. You break them open and add buffers and things. So the random things that you rely on, random collisions and things like that are much more dilute. And also, if you're thinking about this from a sort of understanding biology standpoint or trying to build a cell, this dilution can also influence what you learn about a system. And what we find particularly with eukaryotic systems is that cells are really super highly evolved. So actually, there are a lot of stress response pathways that will shut down the machinery during the lysis procedure. So the cell can tell that things are breaking down, that the cell wall is being destroyed, and that things are becoming more dilute. And so things like phosphorylation events that prohibit protein synthesis can happen. And so if you want to reactivate that or to keep it active, you have to think a little bit about how you do the lysis, which is something that I'll talk about in the talk today. So in case there's anyone who's not aware, there are three components to cell-free protein synthesis. So there's the cell lysate, which has all this lovely machinery. There's the DNA template, which encodes for the protein that you want to make. And then there's the reaction mix, which provides all of the small molecules that you would normally have metabolism for. So things like amino acids and NTPs. But also in the systems that we use T7 RNA polymerase as an orthogonal polymerase. I'm going to talk about eukaryotes, and in particular, when you're making these lysates, the nucleus tends to spin out. So any of the processes that happen in the nucleus will no longer be happening in your lysate. So you have to think about how you can compensate for that. And so what we do to enable transcription then is to add the T7 RNA polymerase. Whereas I know with prokaryotic systems like E. coli, you don't have to worry about this. There are also two ways that you can run this. So you can have a coupled reaction or a linked reaction. So basically the difference here would be, do you make RNA in one reaction and then add that RNA for the protein synthesis, or do you do it all in a single linked system? And then there's been a lot of really interesting work on batch versus continuous flow systems. So in a batch reaction, you put it all into your Eppendorf or 96 well plate or whatever you want, and you let sort of the reactions run their course. You might top up feed with small molecules that you are worried about running out, but in general, the things that are there at the beginning of the reaction get used up, the protein gets made, and eventually at some point it's going to self-limit. There's been a really lot of interesting work of continuous flow systems where you use, say, membrane or dialysis systems to continually replenish things that are running out and to remove waste products, particularly inorganic phosphate that might be building up. So these are two things that you have to think about when you're thinking about running your reactions. And then, of course, the final thing is exactly which cell do you use? And so that could be based on the application end goal that you have in mind. So you can use cell lysates, or you can use this pure system that I alluded to before, which is basically purified proteins that perform the same reaction. But if you're interested in special properties of life, there might be organisms of interest that have special metabolism. So, for example, a colleague of mine at the University of Kent, Simon Moore, is really interested in antibiotic synthesis. And so he uses lysates from streptomycetes, where there's a lot of weird and interesting cofactors and post-translational modifications of enzymes that are necessary. And so his reason for using the lysate from that organism in his cell-free reactions is that those capabilities are inherent. So they come with the cell that you choose to make the lysate from. In general, and I've got a little bit on a slide in a second, yields from prokaryotic systems are higher than eukaryotic systems. So the rates of reaction and the overall final yield is higher if you use a bacterial cell than a yeast or a mammalian cell. And then there's also a really interesting sort of use as well that appeals to me as somebody who works with some of these organisms that are a little bit slow to make cell lines from to manufacture proteins, which is the idea of prototyping. So you might decide that it's easier to test in cell-free a bunch of different variants of a protein or a bunch of different genetic constructs, and then only carry on to make stable cell lines. For example, in Chinese hamster ovary cells are the ones that perform well in cell-free. So this is something that we also have looked at doing with the systems that we favor, which are based on Pichia pastoris and Chinese hamster ovary cells. So this is just a summary from a really good review paper. And if anybody's interested in this field and doesn't know much about it, I would definitely point you towards this. Just so the different types of organisms that people have used before and the different types of reactions that they run. So you can see on the far left here, these are various different cells that you can use for your cell lysate system, ranging from simple things like E. coli to tobacco cells, human cells, and other types of interesting things that people would use as model organisms. Bacillus subtilis, my colleague Paul Fremont has worked on as well. And you can see that the yields really span quite a bit. And in general, those with the highest yields, particularly the systems from E. coli and wheat germ extract, are the ones that people have worked on the longest. And so there's been a lot of effort at sort of improving and increasing the yields. And you can see that really illustrated here on the right-hand side, the far right, the number of papers in using a particular system tends to be high for things like E. coli and meat germ. But it really spans all things in between. And then this graphic here in the middle is just a really nice rainbow way of depicting what people use these types of things for. So different applications connecting to different lysates from different cell types on the other side. So a lot of it is around manufacturing and production and making proteins that are hard to make. But there's also nice activities and education and high throughput screening as well. So finally, to get around to really the point of this, so why do we have interest in eukaryotic systems? And I think really for us, again, thinking about manufacturing, it's twofold. So one is that you can retain particular organelles and compartments that you might be interested in. For example, the secretory pathway, so the endoplasmic reticulum and the Golgi apparatus. There's been some really lovely work by Lenio Bio, who manufacture a system based on tobacco at retaining mitochondria and allowing those to power the energy systems within the cell-free reactions. You can think about developing ways of keeping that nucleus in there if you want to study natural transcriptional processes and things like that. So I like to work with prokaryotes because they're nice and easy and fast, and so do all the students, but I think eukaryotes have a lot of bells and whistles that are sometimes very interesting from a biological standpoint and also from functionality. And in particular for the protein manufacturing world, we think a lot about post-translational modifications, and particular glycosylation and linked and O-linked, as well as some other types of modifications that are sometimes necessary for protein function. So it's no good manufacturing a lot of protein if you don't have it in a particularly functional manner, or if it's not folding properly because it's missing key post-translational modifications. So the eukaryotic systems give us a way of sort of emulating that, but in a cell-free reaction. So in case I've enticed any of you to start making your own cell-free system, I kind of wanted to focus this talk on what the nuts and bolts are of making a cell-free system, and just use as context the two eukaryotic systems that we work with most in the lab, and some of the things that we've done along the way to try to increase the yields and get things working well. So the first example is Pichia pastoris, which is a yeast. It's a non-model yeast. It's methylotrophic, which means it can grow on methanol as its sole carbon source. And it's used actually quite a bit in protein manufacturing. So its first use was in manufacturing of single cell protein. So as an alternative, all the way back in the 1970s, people were thinking about how could you make protein more sustainably using microbial fermentation, which is interesting that we've now come full cycle and this is starting to gain a lot of traction again. In the 70s, though, there was actually an oil crisis, which made methanol become actually too expensive to be used as a feedstock for single cell protein for human consumption. So in the 80s, they looked at repurposing it for a recombinant protein expression system to make things like enzymes for industrial use or for therapeutic proteins. One of the things that we really appreciate about Pichia is it grows to really, really high cell density. So ODs of up to 600. So essentially it looks more like a cell paste than it does a culture. And in our thinking, this would not only provide lots of machinery, but also get us to a point where we can get really highly concentrated lysates and maybe avoid some of that problem that I mentioned before with the dilution of things. It's good at making proteins, both intracellularly and secreting them. Most proteins can be made to some extent in Pichia, so it's nice from a protein manufacturing standpoint. And it's relatively easy to manipulate genetically, maybe not as easy as Saccharomyces cerevisiae, but there are a lot of tools out there. And one thing that really caught our eye from a therapeutic protein standpoint is that they're actually humanized end-linked glycosylation strains where they basically replace the natural machinery of the yeast such that you can get the same types of end-linked glycan structures as you would from humans. So in our mind, then, it's a really nice type of cell to work with for protein making, and a cell-free platform would enable us to prototype things as well as potentially do manufacturing, with a caveat that we really need to drive our yields a lot more if we're going to be able to do that. So this is the same schematic from before. So remember, there are basically three things that need to go in there. There's the cell lysate, the DNA template, and the reaction mix. And so when you're setting up a new platform, you've got to make choices on all of these fronts. So from the lysate standpoint, you need to identify a lysis method that will work. Pichia being a yeast, it can be a little bit tricky to get good lysis and release all that machinery. When do you actually harvest the cells? So at mid-exponential phase, they're growing really, really well, but potentially you might want to wait for a high OD so that you have highly concentrated lysates. So how do you balance that? And are there any engineering targets that you want to improve the extract efficiency? And this is a process that's definitely ongoing. From a DNA template standpoint, it's pretty easy to know how to encode the protein that you're interested in, but in particular with the eukaryotic platforms, the initiation of translation, particularly from a 5' end cap, isn't so efficient within the extract. And so we tend to use internal ribosome entry sites or IRAS sequences in order to get around that. And then with the reaction mix, what do you use in that and how do you optimize the conditions relatively easily? So I thought I'd take you through all of this in the Pichia context. So the first thing we did was look at lysis. I mentioned already the cell wall is pretty tough, so we used a huge range of different types of lysis. So a Downs homogenizer, if no one has ever used it, is essentially a glass tube with a pestle that goes in it, and you sort of squish it up and down. It makes kind of a nice whistling noise, and you're essentially grinding the cells into dust and getting them to release the internal contents. Wiper is a chemical lysis method. Zymolase basically and lidocase do the same thing. They start to degrade the cell wall to hopefully make the cells a little bit more fragile, and then you can combine that with one of the other lysis methods. And then, of course, everyone's favorite methods of breaking cells open, sonication, and high pressure homogenization under different types. So in this figure, the blue bars are the remaining colony forming units per 100 microliters of the lysis solution, which refers to the y-axis that's on the left-hand side. And then the black dots go with the protein concentration that was released. And work from prokaryotic systems suggested that the higher you had protein concentration, the better. And so we were looking for a combination of high protein and hopefully really a lot of lysis. And so you can immediately see that one of the things that stands out here is that high pressure homogenization gives you very high concentrated lysis. So we focused on that initially. I will say that COVID threw a bit of a spanner in the works. So in the first work from this that we published, we were using high pressure homogenization, but when we shut down, that's in another building. And so we have had to resort to using sonication for a period of time in there. And it does still work. It just gives you a lot lower concentration in lysate. Okay, so then we moved on to think about harvesting time. So Mike Jewett's lab had done a lot of work with Saccharomyces and cell-free protein synthesis from that. And they basically were looking at the switch to production of ethanol. But Pichia is actually a Crabtree negative yeast, so it doesn't make very much ethanol. So that didn't quite work for us. So the postdoc who was in my group at that time thought about trying to track what was going on with ribosomes. And one of the other hats that we wear as a group is we look at whole cell biosensors, genetically encoded biosensors. And so she decided to kind of combine the two concepts and come up with what was essentially a biosensor or a genetic reporter for activation of ribosomal genes. So essentially, she took a series of promoters responsible for the transcription of either ribosomal proteins or maturation factors, fused that to GFP, integrated it into the genome at the same locus as the natural one, and tried to look for when are GFP concentrations high, indicating high transcription of ribosome genes, which should reflect high ribosome content. So she made a bunch of these strains, and the first thing we looked at was what happens if you use different media. So in YPD, which is a rich medium, versus NM, which is minimal methanol, you'd expect higher ribosomal synthesis, and you can see that some of the biosensors, especially this blue one here, which is a small subunit gene, reflect that. So you've got relatively high GFP fluorescence in the YPD medium compared to the minimal methanol medium. And so that kind of gave us some confidence that this works to some extent or another. So what we did after that was then culture these across time and look at the correlation between the GFP at different ODs. And essentially what I've got here are data from 4 separate biosensors, each comprising a different strain across OD on the x-axis and the GFP fluorescence on the y-axis. And the difference between the left and the right is just that I eliminated this one here that gives a really high signal, so you can see a little bit better what's going on. So you can see that some of these are more useful than others. Like this one, for example, has a huge dynamic range. It makes it a lot easier to see what's going on in terms of fluorescence. But what we found is that transcription sort of peaks and then sort of either levels off or even maybe to some extent declines as OD goes on. So it suggests that this isn't a strategy that will necessarily tell you too much about ribosome levels. But what we did do is sort of say, okay, it's suggesting that there's not much difference between our harvesting at an OD of 10 or 15 versus an OD of 18 or 20. So we started harvesting it at these higher ODs from that. So that was sort of point one. And then from there, we thought about our DNA template design. And I mentioned that it's a bit more complicated in eukaryotes than it is in prokaryotes. So in prokaryotes, you put your Shannon-Delgado sequence right in front of your start codon and you're sort of good to go. And there's even nice ways of characterizing or quantifying translation rates from different sequences and things like that. With eukaryotes, the natural way of translation for most RNA molecules is that there's a chemical structure at the 5' end of the RNA called a 5' cap. And that's what brings together the initiation complex, and then it starts scanning for the first start codon that it comes to and gets ready to go. So the rate of capping in the cell-free reactions is pretty low. And we also are getting rid of the nucleus, so the capping enzymes are also mostly being spun out of the extract when we centrifuge. So we had to look at using different internal ribosome entry sites, which are basically virus-dependent sequences that help a virus hijack the protein translation machinery of the cell that they're invading by fooling the cell basically into initiating translation at a very strong secondary structure in the sequence. And you can see there are various types here. We screened a couple of different ones in Pichia. So this cricket paralysis virus one seems to be broadly host specific, but then there are some other ones that we also looked at. And essentially what we did was we just basically compared across them. In this case, the cricket paralysis virus that I mentioned a minute ago, which is from a virus that invades crickets, but seems to work in insect cells, in yeast, and even in mammalian cells to some extent. We can use it in CHO. Works pretty well compared to this GP1, which is essentially these yellow squares here, which was something that people had used in whole cells in Pichia before, but didn't seem to work in our cell-free extract. And I don't know if that has something to do with the way that the RNA is folding when it's being produced in this environment, which is pretty artificial, or something else. One thing I want to point out, because it becomes important later, is that we tend to use a luciferase reporter in this system because we have issues with folding of GFP. So these reactions are run static without any shaking, and GFP doesn't mature that well under that kind of environment. So we tend to use luciferase as our reporter instead of GFP. Okay, so we'd identified an IRAS and we identified a harvest point, but we were thinking about some improvements that we could make. And one thing we really wanted to do was go back to this idea of ribosome content. So, I mean, this is the machinery that we're really after. This is the one that's making the proteins. And so we wanted to see could we actually augment the ribosome content of our strains by overexpressing some things. Now, a ribosome is far too complicated for you to overexpress every single protein without causing the cells basically to be stressed and unable to kind of meet the metabolic burden of that. So what we looked at were global regulators of ribosome biosynthesis, so transcription factors, which are controlling the transcription of many genes, or maturation factors. And what we did was something relatively simple. We picked out a few of these, 11 in total, and we just overexpressed them with a constitutive promoter, PGAP, which is pretty strong. And we screen them in the context of a ribosome biosensor strain to kind of say, does overexpression of this give us a higher GFP content, which would imply a higher ribosome content from that. And you can see from this that we had a couple of candidates, FHL1 and HMO1, which gave us higher GFP fluorescence statistically compared to the wild type. We had a couple which are also starred, which were statistically worse. So something about overexpressing this protein gives us lower GFP content, possibly due to imbalance of machinery or something like that. And this is all in comparison to our biosensor strain YTM1 here. So we went on and made extracts of the FHL1 and HMO1 strains. First, we checked the GFP fluorescence, and what we found was that we did have higher GFP fluorescence, and that seemed to be sustained across different ODs. And then we went ahead and made the extracts, and what we found was that the FHL1 strain actually increases the synthesis of the luciferase reporter reasonably substantially by about a factor of 5, which is something that was really good because we were starting out with relatively low amounts of protein synthesis, and that's independent of the IRES, although it's quite hard to see here. So you can see the yellow diamonds higher than the gray diamonds, and the yellow squares are higher than the gray squares, because the IRES actually is important to have. In there, the HMO1 strain actually had pretty low protein synthesis, so overexpressing that seems to do something else to the cell that makes them unhappy in the protein synthesis kind of context. So moving on from there, so we've done all this work with luciferase. We thought, let's try to make a real protein that somebody might care about, and for this we looked at human serum albumin, which is a nice protein to make from a biopharmaceutical context because it tends to be a fusion partner for a lot of the therapeutic proteins. And we just wanted to check that overexpressing the FHL1 also similarly helps with that, and it's not a luciferase dependent effect. And you can see that in both cases, the yellow here, which is the strain that overexpresses FHL1, is higher than the gray, which is the strain that doesn't. And then we kind of turned our attention to the reaction mix optimization, thinking that a lot of work had shown that this was a key factor in the yields in protein synthesis reactions. So what could we do to sort of improve that? So here's where the need to use the luciferase was kind of problematic for us because it involves a lot of handling. So you have to actually sample from the Selffree reaction and mix that into a buffer containing the luciferin substrate and then monitor that over time. So it's not that compatible with things like plate readers doing continuous measuring. It's not that compatible with small volume acoustic liquid handlers where you would ideally be able to use GFP as a reporter for that. So we looked around, and there are actually some nice papers where design of experiment screens have been done using a limited number of samples. I mean, I think I personally had always thought of design of experiments as something where you do like a massively high throughput, several thousand factor screen, and you build models that optimize and things like that. But there's actually a way of doing design of experiments called a definitive screening design, which is essentially designed for this sort of resource limitation or compact designs in mind. So essentially what you do first is you first try to select components and determine upper and lower bounds for these components individually in a one factor at a time approach. And then the design will give you a limited number of reactions to do for the actual model building and the optimization. So the first thing we did was took the components of our reaction mix, which are listed in this table, and we looked just to see, based on previous literature, what are things that people have shown have a strong correlation with cell-free protein synthesis yield. Those are the ones that are highlighted in yellow. Believe it or not, the buffering system turns out to be quite important in literature if you're looking at it. And then obviously things like potassium magnesium balance, the amount of amino acids you have and the amount of NTPs and energy source in there. And so then you look for your upper and lower bounds. And in this phase, what you need to do is identify bounds where you don't have any failed reactions, because failed reactions are not very informative when you're trying to build the model. So you're looking for upper and lower bounds where you see a difference between that and the midpoint, but where you don't totally break the protein synthesis reaction. And in screening individual factors, what you might find are things that go through a clear optimum, things that have a linear relationship, et cetera, or it's not on here, but there are things where you just have a flat line where it doesn't matter how much of that particular thing in a one factor at a time approach you've got, it's pretty much flat in terms of its expression. So Alex Spice, who was the postdoc who did this work, started out by screening the factors, and then he went through and basically in a set of 22 reactions was able to fit a definitive screening model. And so here are just some random conditions. They're not secret. I'm just putting the number here because it doesn't really matter what the condition is. And you see you get a range of activities, which helps you fit a nice model. And from this, we were able to predict a set of optimized conditions that actually gave an increase in protein synthesis. Again, another factor of maybe 3.5 or 4-fold compared to the midpoint, which was our original starting reaction. So once again, we wanted to be sure that it wasn't just a luciferase effect, and we went back to HSA, and again, we see about a similar factor of improvement from that. Okay, so that's kind of the two big things then. We have an IRES, we've got a lysis procedure, and we've got a reaction mix. We went on to sort of try to challenge this, and Alex in his PhD was looking at the synthesis of vaccines. And so he started investigating virus-like particles, which are basically really nice as a vaccine because they're structurally very similar to viruses, but they don't have a genome, and so you don't have to worry about infectivity or anything like that. And this kind of repeat structure gives you a nice immune response. It's quite challenging as a protein to express. So we were interested to see, can you make these in cell-free? And then we were also interested in the fact that because it's an open system, is there a way to sort of identify conditions that would allow you to assemble the VLP, so sort of in a high throughput manner if you're trying to screen in there. So one of the examples that Alex chose for his PhD was hepatitis B core antigen, which forms a VLP, but it's also a nice sort of display system as well. So you can fuse a snippet of an antigen of interest into this, and it will display it on the surface particle. So here's a nice looking thing of what it should look like. And we were developing a way to sort of simultaneously express, or not simultaneously, but in a two-step process, express and then semi-purify and assemble the VLP. The structure of the protein domain for the core antigen looks like this. So at the N-terminal domain has been shown to be enough for the VLP assembly. So if you express that alone, if you get to higher concentrations, you'll start to get particle formation. And there's some interesting things in cell-based processes where you can see these sort of forming inside cells using electron microscopy and things like that. To be honest, our cell-free protein synthesis is not that productive. So if we express just the N-terminal domain, we're able to make the monomer, but we didn't get VLP assembly. But what we found was some nice work where they'd used rabbit reticulocyte lysates and found that actually in the C-terminal domain, there's a phosphorylation event that happens here on a serine residue that actually can modulate the assembly. And by removing the phosphate group, you're able to get assembly at lower concentrations of the core antigen protein. And so what we decided to do is emulate that using just alkaline phosphatase, like the kind that we use for cloning DNA in our lab. So the procedure that Alex drew up looks like this. So he runs his reaction for 3 hours at room temperature. He changes the buffer conditions and adds alkaline phosphatase in what he calls his assembly reaction mixture, incubates that overnight. We're not sure it really needs to be overnight, but as you know, many things in the lab end up being incubated overnight just for workflow reasons at 37. And then concentrate that using a centrifugal concentrator, which you can find for relatively small volumes because the VLP particles should be quite large. Buffer exchange it, and then have a look at what the VLPs look like. The first thing Alex did was run a DLS to kind of see, is he getting particle assembly? And these are, I'm not sure if you can see the individual lines because they're kind of faint also on my screen as well, but you can see that there's a peak here around a size of 38 nanometers, which is around the size of a previously reported VLP that was made in cells. There's some stuff up here of high molecular weight, which is probably leftover proteins that are precipitating out or aggregating together. That we're not really sure what is. So you wouldn't necessarily want to inject this into a human, but it definitely is showing that you can get particle assembly in situ without having to do a lot of steps like gradient centrifugation or anything like that. And then he ran electron microscopy using the facility at Imperial, and you can see that he does get particle formation. So these arrows are pointing to VLPs that are formed. Around 30 nanometers. They look a little bit smaller on cryo-EM than they do in solution in DLS, but this is consistent with what's known in the literature. So we are able to do this, and now you can imagine setting this up as a screening platform where you can fuse in antigens of your choice and look at things like, does it form on DLS? Maybe you want to do high throughput EM, maybe not, but look at different variations, different truncations, and see which things fold well in a nice high throughput manner before deciding which ones to make in your cell-based system. So with that, I'm going to leave the PICHIA story because I know this is being recorded. We're continuing to work on it, but other data is not yet published. I'm happy to answer questions about it one-on-one or afterwards as well. and just kind of dip in a little bit on Chinese hamster ovary cells. For those of you who are interested in mammalian cells as well, it is possible to make cell-free systems from this. We're not the first. Stefan Kubik's group at the Fraunhofer has shown that this is a useful platform to work with. And again, it's one of those things where as protein manufacturing interest grows, Chinese hamster ovary cells are used for about 70% of therapeutic glycoprotein production in cell-based processes. So you can imagine having this ability to prototype. And here's a hamster. These cells actually grow in suspension. So they were smuggled out of China in the 1950s, and basically they are very genetically plastic, which is a nice way of saying that they mutate quite a lot. And so you can sort of train them to do all sorts of things. You can make oxytrophs just by subculturing them. You can wean them off of serum, which is good in terms of manufacturing if you don't want animal components in there. You can actually force them to grow in suspension just by shaking them and not letting them sort of rest down on a surface and grow adherently. So it's quite nice from a manufacturing standpoint, from a cell-based process, but also for a cell-free process to not have to worry about having lots of tissue culture flasks and trying to pool things and stuff like that. So it's been nice for us. I like to think of them as the microbes of the mammalian world. So they're a lot more robust than many mammalian cell types. So I have friends who do PhDs with cells that if you look at them slightly funny, then they'll die in tissue culture and you have to spend the next 3 weeks reviving them and passaging them till you can do your next experiment. These grow pretty robustly. The main thing you worry about is contaminating them with bacteria, but beyond that, if you've got good aseptic technique, everything works really well. They're also able to do post-translational modifications like glycosylation. And here we were focused a lot on the rapid prototyping because the industrial way of making cell lines involves random integration and multi-copies, and then a big sort of phase of screening to look for highly productive cell lines. So you probably only want to invest that time and effort if you're pretty sure about the construct you want to make. And as I said, the Kubik lab had established basic principles, so we were just picking up from where they left off. So again, same graphic. So how do we make lysis? When do we harvest them? Which IRESs do we use? What things do we use in the reaction mix? And here I'll touch on some additional factors for a more complicated host with more complicated biology that we think are necessary. So the first thing we did was we looked at different available commercial growth mediums. So these are all chemically defined medium because that's what's needed for manufacturing, but they actually vary in their composition. And what we found was that just comparing 3 different types of medium, that the cells seem to retain their viability the longest in something called CD-CHO, which stands for chemically defined CHO, followed by this XL medium. This particular cell line in the high clone medium, the density, the viability tends to go down after about day 3, so wasn't the best choice for us. So we focused in on the CD-CHO because it was also giving us relatively high cell viability, density, which I'm not showing in this graph. And then we started to think about how could we culture them. And one of the things that we found was that although they are suspension adapted, which means they should freely sit fine while shaking, they do tend to stick to each other if they can find each other and to the sides of the flask. So this is just an Erlenmeyer flask made out of tissue culture plastic, but you can see the cells are sort of stuck there. And the reason this was a problem was because when we went to lyse them, it made it quite difficult to sort of really break the cells open, especially the ones that were forming a clump, and the ones that were kind of on the inside of the clump were a little protected from things like mechanical or chemical lysis. So one of the things that we did quite early on was we switched from CHO-K1, which is kind of a generic suspension adapted CHO cell line that you can get from LGC standards, to something called CHO-S, which is marketed by Invitrogen, who's now part of Thermo Fisher Scientific, which is a little bit better at being suspension adapted. And then we also found that if you add a bit of antifoam, which is what you would use if you're growing these things in a bioreactor, it's fine. The cells don't stick together as much, and it doesn't seem to harm their physiology in terms of the extracts. We then went on to try a bunch of different lysis methods, so quite similar to the other one. We looked at freeze thaw, nitrogen cavitation, sonication, things like that. Sadly, the method that we've ended up with is passaging things through progressively smaller needles. So you can imagine this is a bit of a chore for the people trying to do this. Essentially, they're whizzing them up and down through a needle method, and basically, as enough cells lyse, you go down to a smaller needle and whiz them up and down a bit more, and do this all in a cold room while it's freezing. But it seems to be the best in terms of reproducibility and getting really good lysis and lysates that actually work. Because for a lot of things, like for example, sonication, we must be denaturing a key protein and it's not active there. So we've got a needle method going. I know some other labs, for example, the Fremont group at Imperial tends to use nitrogen cavitation instead. So I think that there are some other possibilities here. We might want to revisit this if we think about scale up. This is just an example of what it looks like as you're lysing the cell. So here's the culture when you start. Here's after passages through the first needle. You can see there's some cell debris, but also some intact cells. And here's toward the end when you're on sort of the third set of needles, which have the smallest gauge. Almost all of the intact cells are gone, and you can see bits of cell wall left, and the proteins have mostly been released. We then thought about the harvest time challenge. So again, there's nothing like ethanol that we could think of for a good indicator, but what we thought was that maybe we could look at the total ATP and GTP content of the cells as a marker for how energetically healthy they were. And what we found was that if you look at cells in early, mid, and late log phase, so this is days 3, 4, and 5, that they have a higher ATP content on day 4. And the GTP content is similar between days 4 and 5. So we decided to highlight day 4 as a time to harvest the cells. And indeed, actually, if you make your lysate from day 4, you get slightly higher activity in terms of GFP synthesis than if it's early or late log phase. Again, we started looking at the IRES question. So we need to be able to efficiently initiate translation in the absence of the 5' cap. So here we compared 2 IRESs. So EMCV is from a cardiomyocyte virus. And this is what is used in a lot of the commercial mammalian cell-free synthesis kits. And CRPV is our famous cricket paralysis virus, as before. So we found a little bit of a interdependency between the reaction mix conditions and the IRES performance. So if you use the EMCV virus IRES, you have to put DTT in the reaction, which is what's shown here in this light gray reaction. Otherwise, you get very low protein synthesis. But with Crickett's paralysis virus, it's actually the opposite. So the light gray bar here is a reaction mix without any DTT in it. I'm not sure why DTT would affect the functioning or folding of an IRES, which is an RNA structure, not a protein, but it must have something to do with which proteins are sort of oxidized and reduced in different things. It's not something we've looked at in any great detail. So if anybody's interested in this sort of thing or has ideas, I'd be happy to hear them. We tended to use the EMCV virus because of additional protein factors that can increase yields, which I'll talk about in a minute. But just sort of a warning, if you're thinking about building one of these systems, you might have these weird effects that no one could predict from the start time. Okay, so what we found was that even in our quote unquote optimized system or initial optimization, we did have pretty low protein synthesis. And what had been reported in the literature for HeLa cells, which are a different type of cells, was that you can see evidence of some of the initiation factors being phosphorylated during the preparation of the lysate, and this translates to inactive protein machinery. And in particular, people have focused in literature on EIF2, which is one of the initiation factors, and its phosphorylation state. Essentially, there's this set of pathways which basically integrate stress responses of different types. So things like viral, heat shock, oxidative stress, nutrient limitation, endoplasmic reticulum folding stress, and essentially all of the outcomes of the signaling pathways are to phosphorylate this EIF2 protein and make it basically inactive so that it can't initiate protein synthesis very well, which sort of stalls any translation in the cell until the conditions can be improved by other factors and allows various responses to sort of take hold and alleviate stress signaling first. So we think that this is being activated, and there was some evidence from literature as well in human cells that was being activated during the preparation of cell lysates, because again, we're squishing them up and down in needles is probably not the most normal state of being for the cells. So we looked in the literature, and there are a couple of ways that people have combated this. So you can express various proteins. So GAD34 is a phosphatase that essentially recognizes the phosphorylated version of this substrate and dephosphorylates it so that you return to your normal initiation factor that can initiate protein synthesis. There's a viral protein called K3L that a group in Japan had expressed, which is essentially a pseudo substrate. So it can act in lieu of EIF2 as a substrate for some of these signaling pathways, and therefore fools or takes up some of this phosphorylation that would happen. And then a recent paper that came out where they were putting kinase inhibitors in, again, with the idea of stopping this phosphorylation event. So these are three things that we've tried from there. So the GAD34 and K3L proteins, the authors of the original paper where they'd done this in HeLa and hybridoma cells, respectively, were kind enough to send us their constructs for expression in E. coli and either histag purification or histag and anion exchange chromatography purification. We were able to express and purify these. They're kind of pure. You can see this one's maybe got some other stuff in it. The K3L is a bit more pure, and we dope these into the reaction, and we can see that there is an effect of expressing it, primarily down to the GAD34. So increasing concentrations of GAD34 in these three different gray bars here give us an increased yield of GFP in the reactions compared to not adding any accessory proteins at all. And if we add K3L on top of that, it doesn't really seem to help. And if you think about the mechanisms there, it's probably much better to have an enzyme around that can continuously dephosphorylate a protein rather than something that acts as a pseudosubstrate, but once its phosphorylation sites are occupied, is sort of like done its job and there's nothing more it can further do. So the GAD34 seems to be a bit better. We also thought about trying to figure out if we could express these in the cells before making the extract. And I had mentioned to you before that one of the annoying things about working with Chinese hamster ovary cells is that making a stable cell line takes quite a while. So we looked at transient expression. So can we put the vector in the cells, have them express it, and then basically not integrate it into the genome, but just express from the plasmid, and then we harvest the lysate and use it. And it seems to work as well. So the workflow here is a little bit weird in that you're sort of culturing the cells, you add the plasmid with PEI, you culture them a bit more, and then you harvest them, but it is a way of doing that. It doesn't increase yield quite as much as the one where you add it as a purified protein, but it's a lot easier than doing a whole E. coli culture and purifying the protein. So it's an option if it's something that you're interested in trying yourself. But what we have found most recently is that actually this 2-amino purine, which is something that you can buy, works just as well as the GAD34 addition. And so actually, if it's a trade-off between buying something and spending money, and somebody spending a whole lot of their time doing it, I'm quite happy to spend the money. So we tend to use this 2-amino purine instead, because it's a lot easier, and it's our preferred method going forward. And again, these are all aimed at shutting down these stress responses that happen when you're harvesting the cells. So with that, I'm at the end. So what I wanted to do is give you a flavor of what it's like if you want to set up one of these systems from your organism of choice. So essentially the things you need to do are figure out how to break your cells open, preserving the machinery, and maybe think about adding these accessory proteins or 2-amino purine to preserve that. Find a template of your choice, and then figure out your reaction mix components. I would say, it's relatively easy to get something off the ground and running, and then a lot of time is spent more on the optimization. So trying to think about how you can squeeze a bit more protein out. None of these systems that I've talked about today are anywhere near the productivity of the cell-based systems. So they're not going to supplant CHO manufacturing or PICIA manufacturing in whole cells anytime soon. But they do give us a way of sort of prototyping things and figuring out what we might want to invest our time in developing cell-based processes. I think some of the things that we've learned are relatively host-independent ways of increasing yield. So, if you think about it from a broad perspective, you can think about overexpressing genes that are either a bottleneck in the cell, like in the ribosome example, or interfering with the stress response, like in the transient expression of the GAD34 example that I talked about. You can use DOE to optimize the reaction mix, and it doesn't mean that you have to do 10,000 reactions in order to fit a DOE model. Definitive screening designs work pretty well. And it might be that you need to explore the reaction environments for your protein of interest and for specific contexts. And here I'm thinking about the example with the IRAs and the mammalian cells, where one IRAs, you need DTT in the reaction mix, and the other IRAs, you should leave it out. But again, this is all about sort of optimizing, and I think you'd see a bit of activity regardless of the way that you chose to do things, and that's enough to get you off the ground with these sorts of systems. So with that, I'll just thank everyone who's been involved, a lot of different students and postdocs. Rochelle and Alex, I talked about their work. This is actually quite an old picture. Rochelle's now in the Jewett lab at Northwestern, and Alex works in a microfluidic company. And then working on the CHO cells, Ellie and Oscar Marshall have been instrumental in that. I'd also like to thank Simon Moore, who I think a lot of you might know because I think he's active in the Build a Cell community. He gave us lots of advice on how to start these systems off, and funders, and I'm happy to take any questions.
There are a couple questions in the chat already, and just for the recording, I'll have you read them out loud before you answer them, and there might be a couple more rolling in. And then again, to anybody else, if they want to unmute themselves and ask questions, go for it.
Okay, I'll leave the slides just in case I need to go back. It seems weird to me because I feel like I'm talking to myself, but I think you can see both me and the slides, so I'm opening the chat window but leaving the slides there in case. So question from Tanner Muller, for E. coli extract, do most people use Rosetta, or is there more variability in strains for extract production? So I think there's been a couple of different groups who've looked at comparing extracts. A lot of people do use Rosetta, especially if they're going to express proteins where the codon usage might be different from E. coli's natural codon usage. There's also been some really strong work that has been pointing towards using something called BL21 star, which actually has RNase knockouts, which should preserve RNA in the system longer, and the longer the RNA is in the system, the more the more the protein that you should get. And then I think there are other strains that other people have used as well, because they've found them to be as good or better for certain specific proteins or certain specific metabolites that they were trying to use. So for example, if they were trying to use carbon sources that weren't the creatine phosphate creatine kinase system that we're using, some other base strains give you a better platform to start working from because they express more of the natural enzymes in those. And then from Justin Kipnis, in eukaryotic cell-free systems, do membraneless compartments stay intact? Can they be made to? So this is a really interesting question, and something that we're looking at now because we're trying to keep the ER and Golgi fraction for the glycosylation. So really big things like the ER, which is essentially also attached to the nucleus, and a lot of compartments do tend to fragment, especially, well, depending what kind of lysis method you're using, but especially if you use something like sonication, which kind of promotes fragmentation in there. I think I don't know too much about how LenioBio make the extracts from the TY tobacco cells, but they are able to keep the mitochondrion intact. And that's a pretty big compartment. So I think there must be ways to keep them intact if you want to. So maybe using something a little bit less mechanical and maybe a bit more sort of chemical targeted at the cell wall would work for something like that. So I think it's possible to keep at least some of the compartments intact and functional as well. And then from Henry Durball, could you please mention a little about cell-free lysate systems from extremophiles? Would it be possible to have an extract that tolerates high or low temperature, pH, etc., if that extract was made from an extremophile? So there is some work in this area. So I think there have been some geobacillus papers, for example, Dinococcus, I think there's been some work on as well. So yes, I think it's possible to make cell-free lysate systems from extremophiles. I think I'm definitely not an extremophile biologist, but one thing that I foresee is that some of the ways that extremophiles survive the environment is not necessarily fully by their internal machinery being more resistant, especially if you're thinking about things like salt or pH, but rather having good membrane systems for expelling excess acid or base, or keeping salts out, or maintaining osmotic pressure in the face of a high salt environment. So I think there are some sort of questions there about how resistant extremophiles would be to certain types of things. But for example, for temperature, I think there's definitely been some thermophilic systems that most of the enzymatic machinery is adapted to high temperature. Ribosomes are adapted to high temperature and things like that. So I think it's definitely a fruitful area for you to look at if that's your interest. Lynn Rothschild asks, is the high pressure method a French press? Have you tried grinding in liquid nitrogen? It's a nuisance, but it works well for a lot of usage. Yes, so high pressure is French press. I think some people have also used microfluidizers, which are a bit more kind of continuous. But yes, the one that we use is a French press. We haven't tried grinding in liquid nitrogen. I remember doing that when I was a postdoc in Exeter working on Arabidopsis, not for Selffree reasons, but for something entirely different. And I do remember it working really, really well. We were using it for RNA extractions. So it is something potentially worth looking at, and potentially something automatable, right? I mean, we did it with like a same kind of mortar and pestle you'd make pesto with, but I think there are probably ways that you could do it in a bit more automatable way. So I think that's something definitely. Thanks for the tip. From Berta, speaking of nitrogen, what about nitrogen bomb pressure lysis? That would probably be very gentle on ribosomes. Have you ever tried it? So I think nitrogen bomb and nitrogen cavitation are similar. I might be wrong about that. So yes, we have never done it ourselves, but the Fremont group here at Imperial uses it. And the Kubik lab, I think, also has used it specifically with mammalian cells and insect cells. And it's definitely very gentle on ribosomes and probably a lot less annoying than the needles, for sure. Okay, from Eva Samuel, thank you for your talk. Can you please comment on efforts making extracts more long working, for example, some feeding systems that would allow several days of reaction continuously? Yes, so I think this is, from a cost standpoint, if you're thinking about it for manufacturing, definitely a direction to go into. So we have found a very limited lifespan of extracts. So if I speak specifically about the Pichia system, within say 6 hours or so, 5-6 hours, it's usually starting to plateau. Within 8 hours, like nothing really is happening. And actually, if we've got complete protease deactivation, sometimes we actually start to even see protein loss of activity. So the proteins are getting chewed up there. So we tried in the beginning to try to think about what might be running out and just add that. So we tried adding, like, at 5 hours, what happens if we add a bit of amino acid or we add a bit of NTP or stuff like that. And we didn't recover activity from that. So what I think is probably happening is we've got inorganic phosphate accumulation, which can then chelate magnesium, which can kind of then wreak havoc on the biology that's going there. So I think the best way to get around that is to have some kind of a continuous system. So a dialysis membrane, for example. So they make these little devices that essentially, not too dissimilar for what you'd use in structural biology for dialysis of protein, but a lot smaller, because we usually don't have too much in our reaction. They essentially have a membrane on both sides and then just a plastic thing that holds it in place. And you can dip it in a beaker of basically reaction buffer. And what happens is you can get amino acid transport across the membrane, NTP transport, and the inorganic phosphate can come out. So the waste products then are exchanged out. So I think those are a good way of doing it. It is pretty resource intensive in that you have to like dunk the whole thing in a carton of buffer. Another potential thing to think about would be if you could maybe have like a circulating system, so that essentially you could have more of a higher proportion of reaction to the buffer in there that would allow you to kind of replenish things. I'm not sure what the record is for length of these systems operating, but I think you can definitely operate them for a couple of days. And I think some people have reported as much as 10 or even 20-fold increase in yield by using these kinds of systems. So it's definitely worth doing, especially if your goal is to make a lot of protein for the kinds of applications that we're looking at. From Justin Kipnis, in eukaryotic cell-free systems with degraded organelles, what is the effect of mistargeted translation from prokaryotic ribosomes? I'm not sure I fully, so feel free to ask another question as a follow-up if I answer something that you weren't asking, but I think what I'm interpreting is that you are asking what happened. Do you get sort of mispriming with the wrong tRNAs and things like that, which I think happens in non-canonical amino acid systems sometimes. So hopefully that's what you're asking. No.
What I was more asking if you want a specific eukaryotic protein translated on a eukaryotic ribosome, but since you also have these mitochondria and chloroplast ribosomes in the system.
Okay. I got you now. Okay, so in the systems that we use, we tend not to use the mitochondria at all. So that's not necessarily a factor for us. I think in the system that Linear Bio have, where they have the mitochondria, they have mitochondria that are sort of intact. So you would need there to be transport across the mitochondrial membrane in order for the protein to get out if it had been translated in the mitochondria. So I don't think that happens too much. But if you imagine yourself making a lysate and the mitochondria bursts open and you have like different ribosomes and things like that, I think it could definitely affect the yields. One potential way around it would be if you use the endoplasmic reticulum compartment as a microsome and you have a signal peptide, it would take the eukaryotic ribosome sort of starting to translate, docking with that, and the signal recognition particle basically pulling the protein through during translation. So that would kind of almost act as a quality control the same way that it does a little bit within cells. So there might be a way around it from that, but I don't know. There may be work that's measured the kind of proportion of translation by the ribosomes as rough ER versus the ribosomes as mitochondrion, but I'm not aware of the differences or the proportions there. From Hussein, have you looked at the efficiency of your systems in synthesizing functional membrane proteins? So we haven't looked at membrane proteins in either of the two systems that I talked about today. There is definitely some work in the literature where people have looked at that. And I think in both insect and in show, the Kubik group have looked at this, and they have shown that you can get fully formed functional membrane proteins either inserted into vesicular structures that are retained during the extract preparation, like these microsomes I was talking about, the endoplasmic reticulum, or in things like nanodiscs, where you dope those into the Selffree reaction. So I think it is possible to do it, and I think there's actually even work that has shown that you can do it in E. coli as well, if you put the right composition of nanodiscs or lipids or things like that in there to kind of provide a target for that membrane protein to basically be inserted during translation. And I think that was the last one in the chat.
Yeah, it seems like maybe that's the end of the questions. I'll hover a bit and see if anybody wants to toss anything else in there. But thank you so much, Karen. That was a wonderful talk. I learned a lot, and I'm also very excited to go explore this a little bit more in the literature. And thank you everybody else for attending. Again, I'm still hovering if there's any questions, but it seems like you've done an excellent job in answering all of our questions.
Thank you. If anyone does decide to try it and they get stuck or anything, or want to run anything past me, feel free to, I think my email address is on the first slide, but also feel free to send me an email, and I'm happy to, if I can't answer it I'm happy to put you in touch with somebody who can, for sure.
Wonderful. That's fantastic. Okay.